So in this recording, what we want to look at in our final look at the anatomy of a eukaryotic cell is intercellular structures. So remember that inter means between, while intra is internal. So intracellular structures would be structures within one cell. Intercellular structures would be structures between two different cells. Now, looking at eukaryotic cells specifically now, again, we have the cell walls of plants. We're going to look at the extracellular matrix, or the ECM, of animals, and then we're going to look at intercellular junctions to kind of summarize this particular talk. Now, we'll start with the cell walls of plants. You could take an entire course on cell walls when you delve into their complexity. But basically, prokaryotes have cell walls. They're made of peptidoglycan. Fungi have cell walls made of chitin. Protists have cell walls, some do, made of a variety of materials. And then plants have cell walls made up of carbohydrates, right? There's going to be other polysaccharides and proteins embedded in those cell walls. Again, when you get into the details, their structure can become quite complex. But it's an integral component of how plant physiology works. We're going to come back to the idea of cell walls when we talk about osmotic pressure. But it's a great stepping stone when you bring in your biomacromolecules, talk about cell physiology, structures of plants, and plant anatomy. Now, the ECM, or extracellular matrix, is the general term we use for all the stuff that we see in between animal cells in a multicellular organism. So this ECM is made up of glycoproteins, so it's going to be proteins and carbohydrates, collagen, proteoglycans, and fibronectin. They're going to connect to the plasma membrane through proteins we call integrins. And that's really interesting because the integrins can actually act as channels, which we'll talk about later, that respond to stress. So literally, mechanical stress on the system will allow those channels to open and close, and that allows then the system to respond on the inside of the cell to what's happening on the outside of the cell. So when we look at specifically that proteoglycan complex, we see that we have a core protein with these carbohydrates branching off from it. So we have this very kind of, it's going to be kind of a mucus-like matrix on the outside of the cells, holding them together, right? So the function of the ECM then is add support to the multicellular organism, adhesion so the cells stay together, allow them to move to a certain extent past each other, and then regulation. And that's regulation of both the interior of the cell and the exterior of the cell. So intercellular junctions then are going to be ways to connect, hold together very specifically different cells. So in plants, we're going to talk about plasmodesmata, and then in animals, we'll talk about tight junctions, desmosomes, and gap junctions. So plasmodesmata, or plasmodesmata, in plants are literally a complex channel between two adjacent cells. So you're going to allow water and small solutes, as well as large proteins and RNAs, to pass from cell to cell, and this is very controlled. Again, you could take a whole course on plasmodesmata at a graduate level. But for here, we just want to note that they are literally it's like a strand of cytoplasm connecting two adjacent plant cells. This is the source of what we call the symplast when we talk about plant physiology, and this kind of one massive group of cytoplasm that is interconnected throughout the plant through plasmodesmata. Right? Now, in animal cells, we have three types of intercellular connections, and those are tight junctions, desmosomes, and gap junctions. So the tight junctions are going to literally kind of weld the two plasma membranes together. And this is going to be important as you build tissues to exclude extracellular fluid from certain parts of the tissue. You want to think about desmosomes as kind of rivets. Like here's like a point of tight connection between two cells, where we can see intermediate filaments playing a role to hold those together. And then finally, gap junctions, which people like to refer to as the plasmodesmata of the animal cell, but in fact they're very, very different, and I'm going to shy away from that. They do provide a very small gap and a continuous link of the plasma membrane between two adjacent animal cells, so they're not found between all animal cells. None of these are found in all tissues, right? So they're going to have a specific role and used in a very specific way to build tissues. But the gap junction will allow very small molecules to pass from cell to cell based on diffusion. So if you get these into your list of cellular components and cellular anatomy, then we can wrap it up and call that a done deal.